All right, so let's take a look at translation. And translation is a synthesis of a protein using the genetic information encoded on a messenger RNA. This is going to take place in the cytoplasm on the ribosomes. All right, and so what we're going to look at next, this is trying to show translation, and this all makes sense in a minute. All right, so the first thing we're going to talk about is what is known as the genetic code. The genetic code translates nucleotides to amino acids. So the sequence of nucleotides in DNA is going to specify the order of the amino acids in a protein. All right? Now, the problem here is there are 20 amino acids and there are only 4 nucleotides. So you can't do it on a one-to-one basis here. So what we use here are codons. And a codon is a sequence of 3 nucleotides that codes for an amino acid. So we see here, if you get the sequence of CUU, well, that's going to code for the amino acid leucine. You get the sequence UCC, that's for serine. Now, these are on, as it says, on the messenger RNA. So that's where these codons are shown. If we look at characteristics of the code, most amino acids have more than 1 codon. So you see leucine has 6 of these, proline 4, serine 4. And so what that does is it helps protect against harmful mutations. So like in alanine here, instead of being it should have been GUC, maybe it got mutated to GCC. Well, you still get the same amino acid, so it's still alanine, so you still get the same protein. All right. Next, each codon has only 1 meaning. Right. So if you get GAA, I believe that's glycine. All right. So that's what you get there. All right. The code does have start and stop codons. So there's 1 start codon, methionine, so AUG. All right. So that means that every protein starts off with methionine. And there are 3 stop codons. So UAG, UAA, UGA. And those are the last codons there. So they stop so that they're at the end. And these do not code for an amino acid. Lastly, the code is universal, so all living organisms use the same code, from the smallest bacterium to the largest animal that's ever lived, which is currently alive today. That would be the blue whale. All right? And so what this does is it suggests all organisms are related by descent. All right. Now, let's take a look at what else is used in translation. So next here is a transfer RNA, or tRNA. And what transfer RNA does is it delivers amino acids to the ribosome. So this is showing a couple of different models of this, and this is what we're going to see, this like cartoon version of it. All right. So at one end of a transfer RNA, it has what is known as an anticodon. So that's what that's trying to show there, is an anticodon. And this is a group of 3 nucleotides that are complementary to a codon on messenger RNA. All right. And so at the other end, it will carry the amino acid that is coded for by the messenger RNA. All right, so now let's take a look at ribosomal RNA. So this is ribosomal RNA. This is a computer-generated model. This is a cartoon version. So ribosomal RNA, or the ribosome, so it's so along with proteins, makes up the ribosome. So ribosomal RNA, also known as rRNA. So it is made of 2 subunits. We have a larger one and a smaller one. All right. And it has a binding site for messenger RNA, which is literally between the 2 subunits. And it has 2 binding sites for transfer RNA. And those binding sites, which we see over here, are going to facilitate complementary base pairing between codons on the messenger RNA and anticodons that are found on the transfer RNA. All right. Let's look at now the steps in translation. All right, so let's go to the next picture here. So here, in chain initiation, the messenger RNA is going to bind to the smaller ribosomal subunit. All right, then the first transfer RNA is going to bind at the start codon. So AUG was the start codon, so there's the anticodon, so it's carrying methionine on the other side. All right. Then the larger ribosomal subunit will attach here. And it'll attach at what is known as the P site, which is one of these binding sites for transfer RNA. The A site is open. The E is just like the exit site. Okay? So, The transfer RNA is at the P site, the A site is open. So if we go to the next picture here, this is trying to show us chain elongation. So here, a new transfer RNA is going to arrive at the A site. The amino acid or amino acids that's on the transfer RNA at the P site are going to bind to that amino acid that is found on the A site. So that elongates a chain by one amino acid. All right? So the next thing that's going to happen here is that the transfer RNA at the P site now no longer has any amino acids on it. And so now it will leave. All right? And what's going to happen here, once it leaves, that ribosome is going to move down the messenger RNA one codon. So these guys were here, it's going to shift over one codon on that messenger RNA. All right? And so that's going to move this transfer RNA, which was at the A site here, now to the P site. Now the A site is open, and so another transfer RNA can arrive there. And this will occur, the hundreds, maybe thousands of times until the entire messenger RNA is finished. All right? And so that gets us to chain termination. So chain elongation will occur until the ribosome gets to a stop codon. All right? And so when it gets there, everything gets released. So the protein then, as you can see here, it says free polypeptide, which can be a protein unto itself or part of a protein. All right? So everything splits apart and we release the protein.